Based on the data from the HERACLES-A trial, we know that HER2 is a marker, a predictive marker of response to metastatic colorectal cancer. In HERACLES-A, we were able to show a 30% response rate with a combination of lapatinib and trastuzumab. So the main question with the HERACLES-B cohort was to test whether an alternative combination of pertuzumab plus trastuzumab DM1 was able to have an anti-tumor efficacy in this very setting of patients with metastatic colorectal cancer. The HERACLES-B trial was a phase 2 single-arm trial in metastatic colorectal cancer refractory to standard treatment. So the sample size was 30 patients in order to be able to assess a response rate that should be as high as 30%. The key findings of HERACLES-B was that the trial did not meet the primary endpoint of response rate, so we observed 3 out of 30 responses, accounting for 10% of response rate. However, we observed in 80% of patients stabilization of disease. So for some reasons, this combination of drugs was not exerting a tumor shrinkage, but rather a disease stabilization. These findings should be taken into account as a possible HER2 targeted strategy for those patients that are more fragile and where you don't need to achieve a tumor shrinkage, since our results are showing that we were able to induce disease stabilization rather than response, and the tolerability was very good.